Bismillahirrahmanirrahim. Today we'll be learning the neuroglial cells and the blood-brain barrier. For this, concentrate from the superior aspect. You find the pia mater and the cerebral cortex, right? But what you see that in between the pia mater, there must be some pial foot processes are there, and they are coming from the underlying neuroglial cells called astrocytes. And what they are forming the glia limitans. And what it is the glial limiting membrane that is present by the expanded termination of the neuroglial cells called astrocytes, right? So present here in between the pia mater and the cerebral cortex, right? After that, we have to look for the cells present over here. Definitely you can observe the neuron here, right? And here you see that various neuroglial cells are present: the astrocytes, the ependymal cells, the microglial, and the oligodendrocytes, right? Another good view to show you all. Let me mark them: the ependymal, the astrocytes, the microglial, and the oligodendrocytes. The oligodendrocytes and astrocytes could be called as macroglial cells, right? Now in this picture you can see the neuroglial cells of the central nervous system and as well as that of the peripheral nervous system. So let's name them again. The ependymal cells in the CNS, oligodendrocytes are there, microglia are also there, and the astrocytes, right? And if you see the list of the peripheral nervous system, you see the Schwann cells below and the Stellus cells above, right? Now, as you know that your neuroglial cells, why they are present? Because they are supporting cells, so they cannot generate the action potential. So what they have to do? They have to provide the mechanical and metabolic support, right? And their number is almost 10 times more than that of neuron, right? But when we stain with H&E technique, we only watch their nuclei. But for the study purpose, we have to see so many schematic diagrams. So let's start with the individual details of all these, right? And before, so for this picture, we should know that neuroglia, we can divide it into neuroglial proper and the ependymal cells. As in the picture, the pink color cells are ependymal cells. Where they present, they basically lines your cavities in the brain and the spinal cord, right? And if we talk about that developmentally, the astrocytes, oligodendrocytes, and ependymal cells, they are coming from ectoderm of your neural crest, right? And what about the microglial cells? They come from the precursor cells present in the bone marrow, right? Now we will discuss them one by one. We have to talk about the astrocytes. They are having the centrally located nucleus with the pale staining because they are showing the vesicular nucleus having euchromatin or finely dispersed chromatin meant to produce protein, you would say. And here you can expect that this is a picture of the protoplasmic astrocyte. So we know that the astrocytes have got two varieties, the protoplasmic astrocytes and other is the fibrous astrocytes, right? The protoplasmic astrocytes, where should be found? They are present in the gray matter of your brain and spinal cord. They usually give rise to the processes, as you can see, which are short, thick, but they branch extensively, right? Because they have to form the dilated perivascular end feet along with your blood capillaries, because they have to take part in the blood-brain barrier, right? And what else we can expect about them is, we talk about a thing earlier that they have to form the glial limiting membrane or glial limitans. And what it is, this membrane which is formed by their end processes expanded subpile feet, they are impermeable relatively, and so around your CNS they create a barrier, right? Now we have to talk about the fibrous astrocyte, how they look like. They are basically present in the white matter, having few processes but their processes could be thinner, straighter, and longer, right? And if we see, their processes can also be showing the terminal ends as perivascular, you would say, feet, right? And as compared to protoplasmic astrocytes, these fibrous astrocytes have got much higher number of intermediate filaments, so that is why they take the name fibrous astrocytes. So we have to now mention the main functions of your astrocytes in general, either your protoplasmic or the fibrous astrocytes. So they have to take part in the transport of oxygen and nutrients. How? Because as you can see in the picture, they are clung to the blood vessel. In this way, they exchange oxygen and waste products with the neurons and in reverse with that of the blood vessels, right? And they have to produce a microenvironment around the neurons as well, because they are supposed to maintain the ionic concentration in the interneuronal compartment. Let me show you the other picture. As you can see, these protoplasmic are nearby the neurons, and this way they maintain the microenvironment, right? And another important thing here, you can see that they are supposed to give the perivascular end feet, and this is clung over your blood capillary to form blood-brain barrier, right? And what it has to do, it has to restrict the movement of various molecules from the blood into the CNS, right? And the processes of astrocytes, when they are extending to the, you would say, basal lamina of the pia mater, they have to form a impermeable membrane. We discussed earlier so many times that they are forming glia limitans, right? And in this way, they surround your brain and spinal cord. And the function of metabolic exchange, which we discussed, that it is present in between the neurons and the capillaries of the CNS. You can see the picture that on one side there lies the neuron, and on the other side there lies the blood capillary. So they control the, you would say, chemical environment around the neuron. So they clear the intercellular spaces of increased potassium ions and neurotransmitters, like can you mention, these could be glutamate. And what else they have to do? They now remove extra glutamate and convert it into glutamine, which can be returned to the neurons, right? And what else they do? They contain or having the glycogen in them, and in this way they release glucose, right? So a very good source of energy metabolism for your CNS, right? So doing a lot many function. And in addition to this, if I mention that astrocytes, especially fibrous astrocyte, they perform the function of formation of scarring tissue in the damaged area of the CNS, right? Now we will discuss the blood-brain barrier a little bit in detail. We can see that there could be the blood-brain barrier, and it is, and you would say, interaction between the glial astrocyte and your capillary endothelial cells, right? And this blood-brain barrier, what it has to do, it protects your neurons and the glia from bacterial toxins, infectious agents, and other exogenous substances. And in this way, they have to maintain a stable composition and a constant balance of ions in the interstitial fluid that is required for the normal neuronal function, right? And before going to the detail that by which they are formed, we have to know that the component of this blood-brain barrier, they are not present in choroid plexus. Why? Because there is CSF production occurring, and another place where this blood-brain barrier is not present in the posterior pituitary. Why in posterior pituitary it is not present? Because hormones are releasing, and in the regions of the hypothalamus where plasma components are being monitored, right? And in the picture you can see the terminal or perivascular foot processes of the astrocytes are clung to the basement membrane of the you would say capillary endothelium, right? And it means this is formed in between the two things. The limiting layer of the perivascular astrocyte feet, you would say. The other thing is the capillary endothelium. And in the left upper corner, you can see that there are tight occluding junctional complexes which are sealing tightly together various capillary endothelial cell, right? And in this way, they are making a strong barrier and saving your CNS from any bacterial, you would say, infection, right? Now we have to talk about that microglial cells, right? They have got rod-shaped nuclei, as you can see in the elongated cells, and they are deeply staining due to condensed chromatin, right? And what else about them? They have no intermediate filaments in them, and but showing the tortuous processes and having small spines over them, right? They occur in the gray matter and the white matter as well. And what they have to do, what is their function? The microglia, they have to perform the phagocytic function, and in this way they are a part of your mononuclear phagocytic system, right? And whenever there is any injury or disease, these cells, what they do, they enlarge, becomes motile, and they act as a scavenger cell and clear the cellular debris. In this way, they can also act as a you would say as an antigen presenting cells, because they have to secrete a number of immunoregulatory cytokines, right? And definitely they are a type of I have mentioned the macrophages, so get their origin from the bone marrow, right? Now look at the astrocytes. They have the rounder and denser nuclei, you can see in the very center, right? Oligo means scanty, right? It means they do not have their perivascular feet, but their cytoplasm definitely filled with the cellular organelles as that of previous cells like mitochondria, Golgi apparatus, but we expect no intermediate filaments in them. But do you know there are two varieties of your oligodendrocytes? One is interfascicular, that is present in between the nerve fibers of the white matter, and they are forming myelin sheath over there, right? And the other variety is the perineuronal oligodendrocytes, and where you get them in the gray matter, right? Closely associated with the cell body of the neurons. And we should remember that astrocytes and oligodendrocytes can be referred as macroglia, right? Now you can see the ependymal cells, which are cuboidal or low columnar. They are showing the microvilli or cilia on them. And where are they present? They are present in your ventricles, right? Ventricles of the brain, you can recall them, and in the central canal of the spinal cord. And they line the or form a layer called ependyma. And they lack the basal lamina. That is important about it. Their cytoplasm is filled with all the organelles, like they are abundantly filled with the mitochondria, Golgi complex, and are well laden with the bundles of intermediate filaments, right? They are showing the tightly bounded junctional complexes in between them, especially near their apices, right? And apices are showing, I mentioned, the microvilli and cilia. Why both things are present? Because microvilli helps in the absorption of CSF, right? And the cilia has to facilitate the movement of CSF, right? Okay, another important thing we have to learn a little bit, that cerebral cortex, if we cut the top gray matter 3 to 4 mm depth, we can see this darker color, the gray matter, right? And in this picture, we have to learn that the structure of cerebral cortex that varies in different parts of it, right? So we have to learn with the two terms. One is neocortex, as you can see, and the other is the allocortex along with the mesocortex. The neocortex is what? It is phylogenetically a new cortex, right? And neopallium word is also used. And it is made up of 6 layers, which we have discussed in the topic of cerebral cortex. And what about that allocortex? This is basically showing 3 to 5 layers of your cerebral cortex and is an old type of cortex, right? Phylogenetically it's being said, and more simpler in structure and is present over the area where hippocampal formation is there, and it is called archipallium over there, right? And when it is present along the olfactory cortex, we call it as paleopallium. These are the few things we have to mention regarding the cerebral cortex. Thank you very much.